Niacin, vitamin B3, nicotinamide riboside (NR), and nicotinamide mononucleotide (NMN) are three biochemically interconnected precursors in the biosynthesis of NAD+, nicotinamide adenine dinucleotide, a molecule essential for energy metabolism, DNA repair, epigenetic regulation, and cellular longevity. While NR and NMN have gained traction in the wellness and longevity industries as premium, designer molecules, niacin remains the only form among the three with a long-established clinical history, extensive human data, and unique mechanistic features. Understanding the biochemical divergence and convergence of these NAD+ precursors, as well as their distinct tissue uptake profiles and metabolic pathways, reveals not only their complementary potential, but also reasserts niacin's often overlooked legitimacy as a potent and scientifically validated therapeutic agent. The NAD+ biosynthetic landscape includes multiple pathways, of which the three dominant ones are the Price-Handler pathway, the salvage pathway, and the de novo pathway from tryptophan. Niacin, nicotinic acid, enters the NAD+ synthesis network via the Price-Handler pathway, which converts it into NAD+ through a relatively direct route involving the enzyme NAPRT, nicotinic acid phosphorybosal transferase. In contrast, both NR and NMN are funneled through the salvage pathway, which recycles nicotinamide, often a byproduct of NAD+, consuming enzymes such as sirtuins and PARPs, back into NAD+. NR is phosphorylated into NMN, which is then converted into NAD+ by NMNAT enzymes. Thus, NR and NMN are more directly linked to the cell's internal mechanisms for maintaining NAD+ levels in the context of cellular stress and aging. One major implication of these differences lies in tissue specificity. Not all tissues express all enzymes equally. For example, the Price-Handler pathway is highly active in the liver and kidneys, where niacin is effectively converted into NAD+. NR and NMN, on the other hand, may be more relevant in tissues with high NMPD activity, nicotinamide phosphorybosal transferase, such as muscle and brain, where the salvage pathway predominates. However, recent studies suggest that NMN may not be readily transported into cells as previously thought and may first be degraded into NR before uptake, effectively making NR the more bioavailable salvage pathway precursor. At the same time, certain tissues like the liver and immune cells still retain strong capacity for niacin-based NAD+ synthesis, reinforcing niacin's potential for systemic NAD+ repletion. Niacin also holds a therapeutic uniqueness not shared by NR or NMN. It has been FDA approved for decades to manage dyslipidemia and cardiovascular disease. Through activation of the GPR109A receptor, also known as HCA2, niacin exerts anti-inflammatory and lipid-lowering effects independent of its NAD+ boosting role. These pleiotropic effects contribute to its broader impact on cardiovascular and metabolic health. In contrast, NR and NMN, while promising in preclinical models for aging, neurodegeneration, and metabolic disorders, lack the same regulatory approval or robust longitudinal human data. They have shown potential in boosting mitochondrial function and enhancing sirtuin activity, but long-term efficacy, safety, and clinical relevance remain under investigation. Another layer of complexity arises from feedback inhibition and methylation demands. All NAD+ precursors eventually elevate nicotinamide, which in high levels inhibits sirtuins. Niacin's slower conversion avoids acute spikes in NEM, reducing this inhibitory risk. Furthermore, methylation of excess NEM to form MANAM consumes methyl groups from the SAM cycle. NR and NMN may contribute to this burden more significantly than niacin when consumed in high doses, though the comparative methylation load remains an area of ongoing inquiry. Despite the excitement around NR and NMN, niacin has often been unjustly relegated to the background due to its flushing side effect. This flush, however, is not merely a nuisance. It reflects pharmacodynamic activity through GPR109A, a receptor with anti-inflammatory benefits. Moreover, flush-free or extended-release formulations allow therapeutic dosing without the discomfort, enabling niacin's full potential in clinical settings. In metabolic syndrome, non-alcoholic fatty liver disease, and even certain cancers, Niacin's ability to modulate NAD+ pools and downstream cellular processes remains unmatched in terms of cost, availability, and scientific grounding. In conclusion, while NR and NMN represent exciting avenues for targeted NAD+ boosting, particularly in aging-related and neurodegenerative research, their emergence should not eclipse the profound scientific and therapeutic foundation of niacin. Each precursor plays a distinct role in the NAD+ ecosystem, shaped by its metabolic pathway, tissue targeting, enzymatic context, and regulatory profile. Rather than viewing these compounds in competition, a more nuanced appreciation reveals their complementary strengths. For individuals or conditions favoring specific tissue targeting or bypassing enzymatic bottlenecks, NR and NMN may offer advantages. Yet for global NAD+ repletion, cost-effective clinical intervention, and systemic metabolic modulation, niacin remains a cornerstone, both historically and scientifically.